Information Science Department at the University of Delaware. I've known Vijay and his work for some time. His earliest work in his thesis at the University of Pennsylvania, which he finished in 1987, was a study of tree adjoining grammars, and was responsible for really taking the very elegant idea of tree adjoining grammars and turning it into a sort of rigorous formalism and showing a lot of very interesting properties that the formalism has. Since then, Vijay has done a real range of work, from more work in defining grammar formalisms to work in natural language generation, work in parsing, work in part-of-speech tagging, super tagging, and the like, and more recently in areas in bioinformatics that we'll hear some about today, in the area of information retrieval and information extraction in the biological domain, as well as more recently in issues about parsing of sequences, base sequences in genetic databases and so on. One interesting thing about Vijay's work has been that he's been interested in a range of questions, both mathematical questions as well as questions of practical interest, and also he's been very interested in, on this I've collaborated with him to some extent, on trying to think about the way these things interact with questions about giving reasonable and linguistic descriptions, and what kinds of roles both formal systems and practical systems can have, and the integration among those things. So it's our pleasure to have him here today to talk about named entity classification in the biology domain. Thank you, Vijay.
Thank you. And thanks for having me.
Well, I don't think this seems any harder.
Well, you can make it bigger by making it show. Right. Yeah. Yes, it also.
Okay. Let me start off by saying that this work is more about investigating certain sources of information that might help us in named entity classification in the biology domain, rather than putting together a system that finally does it. Fairly preliminary stage as far as that is concerned, but rather more about investigating what kind of features might help or might not. So why is there interest, to the extent that already some people are giving a name for this field called bio-NLP? The reason is, in bioinformatics, just about all bioinformatic projects, their success depends on a lot of data being available in computer accessible forms in databases. And even if all the new data that people are coming up with or discovering are put immediately into computer accessible form, it will still be the case that just about a huge proportion of information is there right now only in the form of the literature, residing in the literature, right? So there's been interest to kind of apply language technology tools to take that information which is there in unstructured form in text and put it into more structured form in databases. Okay, and that is also a little feasible because in this domain there's a lot of text available online. There are of course electronic journals online that you can get through libraries. And also in this domain you have something that was started by NIH way back in the 60s called Medline abstracts, and records corresponding to just about all papers in journals and conferences in both biology and medicine are stored in Medline, and there are over 12 million records corresponding to individual papers, and about 7 million of them actually have the abstracts of these papers. So there's a lot of information text that is available.
So what's a record?
It could have a lot of different kinds of information about papers. So it could mention some of the gene names that are appearing there. Some might have just citation information. And about over half of them have, in addition, the abstract.
So they have full articles for subset as well, right?
A much more.
Yeah. Can you go back that little slide?
Sure.
Those numbers.
And then before I really get in, there's a whole lot of possible applications of language processing, right from clustering these abstracts into different topics, built using in order to build better, more intelligent retrieval systems, various text mining applications, knowledge discoveries are very, buzzword that is being mentioned often in this field. But my interests are more in information extraction. In particular, there's a few certain kinds of interactions between proteins that I would like to extract out. And that particular interaction is called phosphorylation. Now, there are three kinds of things that I'm trying to extract for every phosphorylation interaction. Over here, if I see a sentence pattern such as this, A would represent the agent of phosphorylation. What does the phosphorylation? B is what gets phosphorylated, what I'm going to call the target. And C would mean that interaction affected some particular residue or place within the protein sequence. So C is some place in the protein sequence. So if I tried to use standard syntactic patterns to capture this kind of information, we pretty soon realize that that wasn't enough. Here's a typical one, and it works fine here. Sentence. On the other hand, if I look at the next sentence, what appears in this place after phosphorylation is not the target protein, but rather the site within the protein. That is, the target protein is instead specified here. Right? Or another sentence that we found would say, well, in the second position, like in that pattern over there, B, the second argument here, is indeed a protein or a gene, but what comes here in is not a location within the protein where phosphorylation occurs, but rather it's talking about certain cellular location where this occurs. So, for this reason, and for a reason that many other times the arguments are definite noun phrases that you need to do co-reference, we found that for co-reference too, the information about the class is very helpful. Right? For example, this sequence, what does it refer to? If I knew octamer element is also some kind of sequence, then immediately I will be able to say that has more chance of being co-referenced than anything else. Or in this particular case, when I say here it interacts with, I'd like to know what type it is. Or if I knew it was protein, then I could look for proteins before that to do the co-reference. And later on, we'll show how we figure out that this will correspond to proteins from the contextual information. Okay, so because of this reason, our first thing was we started building a named entity recognizer that recognizes these different kinds of types which were important for phosphorylation purposes. We broke the tasks, or at least thought of the task, as two separate tasks: detecting the name and then classifying it. Okay, and for detection, I'm not going to go into detail for detection purposes. Standard methods that we use, such as orthographic information and other information, were used. But also realize there are other kinds of names or terms within names that help for detection. But more than detection, we felt like this really helped for classification purposes. Oftentimes, names in this domain can be pretty long, almost looking like noun phrases describing what these are. Something like simian immunodeficiency virus, or the fact that this is a kinase or a factor, etc. These are names. I can almost think of them as descriptive noun phrases, and in noun phrases in English, which tends to be right branching, the head word often bears the type information. right? So we kind of look at the rightmost. And if you look at all these names, the rightmost word, they kind of tell you what kind of function it serves or what kind of class of entity it is.
So the head word isn't always at the right, like the bacteria that you call line. No, it's where the name called it.
Yeah, I'll talk about that. How we get around that. Like you could have vitamin B. B isn't giving you one.
Yes, exactly.
So we thought this could be used, and we thought of other possible sources of classification, such as context. If I can figure out that MAPK, that what the full form was from text, then I can use the full form to get the type, and so on. Right? But this was very kind of like preliminary work in which, like these terms, the head words that I'm going to call F terms for some certain historical reasons, we don't need to know what F terms stand for, just think of it as a head word. We just had like 30 or 40 of them that we associated with different types. The contextual, we looked like only just a few handful of terms that might help us, such as word expression. If I had something expression, that is probably a gene that is being expressed, or things like that. And we wanted to obviously not rely on hand selection. We wanted to go beyond this, and we wanted to do it in some kind of automated fashion. And that became possible because at the point we did the original work, there wasn't any tagged corpus available, and one became available. And once one became available, we wanted to do this more systematically, look at the contributions of the different sources, and that's what this work is about. So what was the corpus? It's called the Genia corpus, developed at the University of Tokyo, which has 2,000 abstracts corresponding roughly to 70,000 occurrences of names. And realize that's about 35 names in each abstract. Right? Abstracts are not that long, so there's like packed dense with names. And that's important that I'll tell you how that affects us. These names they classified into 36 different types. We didn't use the 36 different types. We combined some things like protein molecule, which we didn't even know from their description of the Genia system what the difference was between protein molecule. And we didn't care whether it was monocellular organism or multicellular organism, so we packed some of these together into 23 different types. Let me just quickly say that we noticed there were like 41% of the names were protein names, 15% approximately were cell names, and 11% is DDOR, stands for DNA domain or region, which could be, for example, a gene name, or could even be part of a gene. Okay? So gene, after all, is a certain region within the DNA sequence. And these are the top three, and there are about 7 or 8 that even are like only 1%. Okay, so as I said, I wanted to find out which of these head words give us information, so I wanted to start looking at these head words, which is rightmost words in names, and say, can I see all the names that, for example, end with kinase, and see, does kinase, all the names, what does that distribution look like? If it's all indicating they are proteins, which is what kinase means, it's a subclass of proteins, then maybe this is very informative, it's going to help us. Now, we couldn't just take the rightmost one because we can have things like vitamin B or tumor necrosis factor alpha, and we wanted to ignore alpha, B numbers, single characters or Greek characters, numbers, etc. We call them specifiers for some reason, and then we ignore that and take the rightmost non-specified word. Okay, and then we started doing this work, as I said, look at each possible candidate F term, look at the distribution and select. If I wanted to, I could select based on some frequency or likelihood threshold. But soon we decided why arbitrarily cut off, take any likelihood threshold, because after all we want to, in the end, combine lots of different sources, so let it be. Maybe the weak ones will never get used. So basically in the end we've started looking at saying, look, take a look at all of these, just consider the distribution and just take that into account. Okay, and we did this for some substitute corpus, and now how do we use that? We started this coming up with decision list rules that might say if, for example, you have kinase as the F term or the head word, then we notice from our corpus that it's definitely always protein, right? If it's clone, then it doesn't have to be. 61% of the time in the training corpus, it's a cell, 29.7% it's DNA, and so on. Right? And then we decided, well, instead of just looking at that, what if we looked at the previous word, sort of like not the head word, but the modifier. Right? And so that's, I've written the template we use. So if that head word is clone and the previous word before that was cell, then I know the chances of it being cell is far higher. It was something like 0.98. And I guess the result was actually noise in the corpus. The final rule says, well, you don't necessarily always have to look to the modifier. You could also look at the specifier, which was to the right, which we'd ignored. And that helps in a rule like this. For example, if I say protein, it doesn't have, if the head word is protein, it doesn't have to be a protein. It could even be a name of a protein family. Okay? However, if that is followed by, here is some Arabic number, if it's followed by a number, then it's definitely a protein. Okay, so we have basically what I could think of as some kind of hierarchical decision list, because all of them had to first be based on F terms, and then sometimes we look at the specifier, sometimes the previous word, or sometimes nothing. Okay?
I'm sorry. I think this is something that you said that I just missed. The entities that you're classifying, there's a stage called bracketing that has to take place first. What are these? What are the boundaries that define an entity? Did you say how that is done?
No, I didn't. I went straight. Like I said, we divide the task into name detection of the entities and then the classification. Okay. Now the detection uses usually lots of orthographic features and certain words and so on, and part of speech tag is used by all. But I'm not in this talk talking about that. I'm saying let me use the Genia corpus, the names and their tags. Let me take that and see how I can classify it. And that's why I clearly know what is the right mode of the head word, right? And I have a couple of things to mention about that quite a little later on. No, the question was, there's a question when you have such overlapping features, how to order those, and I think I'll discuss that a little later. Well, in addition, as we were doing that, we thought, it's not surprising in the scientific domain, that suffixes could also help. In some sense, they are like F terms. Because if something ends with sites, which, the suffix sites, CYTES, I think in Greek it stands for chamber, and it's used for cells. It's sort of like saying, leukocytes is sort of like saying leukocells, right? So cells, if it was a separate word, we call that an F term. And this is essentially playing the same role, such as, for example, even the word blast, which stands for a cell in an immature stage of development, right? And in fact, we notice sometimes Our F terms such as virus and proteins also appear as suffixes, like in lipoproteins, right? Like the good cholesterol, which is high density lipoprotein, right? So that's actually a suffix. Adenovirus is a virus, so why not take? So we decided to try to extract suffixes as well. So how do we do this? Well, we take the head words, right, and then start looking at suffixes of length 3 and see if you need to increase it. And so the way we did it is at any point in time, if you have a suffix of length n, look at its distribution, then compare it with suffixes that extend one more to the left, right, of length n plus 1. If in doing so, you can use standard information gain, any kind of metric. If you see there's a gain in doing this, you go ahead and do it. So if the bigger suffix is more informative, then go ahead, split it, and continue now at this point with the different suffixes. So let me show you a couple of examples. For example, we start with three-letter word suffix ine, and it's really distributed all over the place. There are 384 occurrences in our training corpus, which correspond to cell names. AAM stands for amino acid monomers. There were quite a few of them. Proteins 257 and so on. So now if you take it one more to the left, you see that if the previous one was K, then it's almost exclusively protein. If it was L, it was completely only cell names, right? Because this is like cell line. Okay? And RNA, TINE, these are all seem to be all completely amino acid. So this is a perfect example. If you take one step further, you can almost completely disambiguate it. And so this is a case where we would proceed. We also notice that if I start with YTE, every occurrence of YTE was preceded by the letter C, right? And every occurrence of site was preceded by O, right? So you have examples like leukocyte and monocyte. And the question is, where would we want to go? I mean, in actuality, the correct suffix is site. If I say I can go all the way to oocyte, take the most specific one from this corpus, then I might be overfitting. And actually, rather oddly, even if I went to YTE, I might actually be overfitting here too. And so, this is one where we're not sure where to go. And for us, in this case, it didn't matter. So, we actually got a lot of very good suffixes, over 100 suffixes we were able to pick. And here are a few examples. The suffix, for example, is OMAS. That's a tissue. What does that mean? Well, like lymphoma, sarcoma, and so on, are cancerous malignant tissues. So we were able to extract that. Ending with ZYME is a protein, and so on. So again, we have the distribution. Don't think of this as addition list, making addition, but rather being distribution. So what do the results look like for F terms and suffixes? Well, we put different thresholds, right? So very high threshold, saying, at least 0.9 favoring something. Well, we'll get really high precision. I should have put this in here, except my PowerPoint, I'm not very familiar with PowerPoint, so I wasn't able to add what these axes stood for. So this is precision this way, and recall that, right? Y-axis is recall, and you get 96%, 96 precision with about somewhere in the mid-50s. And then you can keep going down almost to no threshold, and you still have high, the recall doesn't go up that much, and the precision drops to about 90. And this, I found rather surprising that This kind of really emphasized the point that how these names in this domain are descriptive noun phrases, that I could get 60% of these, right, just by looking at the head word. Okay, and that they're really these noun phrases. So what remains unclassified, it depends on what the cutoff was, and in one particular case, the first simian immunodeficiency virus had a box was a name that was not classified because of the frequency threshold. Box didn't appear that often. And then, of course, you have things like, MEK1. If they had defined what the full form of MEK was, then you might have guessed. But since it wasn't defined, then we have nothing, no name internal clue to tell us what this would be. As with STAT1 alpha, STAT is a class of protein families. You have tags, you have all kinds of names that they're so well known that people don't define what they are. Right?
So you could expand the feature space. I mean, the fact that the length of the word, for example, proteins and genes in particular tend to have like 3-character plus maybe a number following it, sort of thing. So a feature saying, is it all caps? A feature saying, is it 3 letters of the, just 3 consecutive letters that don't tend to have high, n-character, n-gram frequency. I mean, there's other features that you could do.
Well, those features are actually very helpful for the detection, not that much for classification. Because a lot of these are like acronym in nature, so it could be any kind of thing.
Okay, yeah, right. So it could be protein or gene.
Right. So it doesn't help that much in classification. It does help quite a lot in detection.
That's what you not.
Do you go outside the domain of, say, one abstract? For example, MEK1 may have been indicated to be an acronym in some other abstract with parentheses around it.
No, but we thought one thing that could help, actually, in some sense, yes. What we thought initially that would help is, what if we had some dictionary of common names, right? And then I can look it up. There, unfortunately, even for proteins, there isn't any exhaustive one, right? And certainly for other types, there are 23 types, we don't have dictionaries. So what we thought was something along the lines of saying, why don't we use the training set as giving us the examples? So we'll find, if not the same thing, at least something similar. Right? So we wanted to look at some kind of similarity in nearest neighbor. So, of course, how do we detect similarity? This thing is not working. They're all this standard string matching, which can be used to define distance measures, right, between pair of names, just did not work. To give an example, if I had the name, I was trying to tag the name simian immunodeficiency virus box, right? And I have an example such as human immunodeficiency virus. Distance-wise, there's a lot in common. But for classification purpose, it's just not relevant. Because that is a virus and this is some kind of box, which actually is a gene part. Right? So what we felt we had to do was, we have to build into our string matching algorithm a bias to the right. Right? Differences in the right have to be penalized heavily, and likewise similarities in the right have to be given. And of course, you also need what we thought was this normalization of names, because people, tend to, don't necessarily get.
It seems like another thing you could do is you could partially or fully parse the noun phrases, and you had trees, and you could try to align these trees among the distances in those trees. So presumably, Tata box would be kind of the head word, and you could try to align the head word with the phrase.
Yeah, thanks. Well, we'd like to try that out. Now, NF kappa B, many authors sometimes just put them all together. Some are going to separate them out. So we just normalize, locate numbers, Greek characters, hyphenations. So we basically normalize so that if you have NF kappa B element, is your query, or as an example, we're going to represent it in this manner, right, with spaces, so that it's clear. Stat 3 beta, sometimes people put them together, sometimes they put stat 3 together, beta separately, and so on. So we do some normalization, and let me show you some examples again in the ones that we found for NF kappa B element. The closest nearest neighbors are all ending with element. And I've shown you how they are aligned. And notice that in, for example, the T delta 7 element, because we look at the types, because kappa is a Greek letter, delta is also a Greek letter, the difference, the penalty is minimal. If you're going to replace a number with a character, maybe a little higher, right? But words with words substitution is penalized heavily. So in this case, for example, NF and IFN, that's a heavy penalty. Okay, and the closest one according to our distance measure for simian immunodeficiency virus had a box, this had a box, and human immunodeficiency virus doesn't even show up anywhere near the top. Actually, we kind of built it in to make sure it doesn't show
I'm
up.
surprised actually that is that supposed to be order from best to worst?
Yeah.
So I'm surprised at the position of the number 2.
But Jason said it's a sequence. DNA sequence string. TATA.
Right, but I
Okay, yes, this is done at the letter level and not at the word level.
some of those matches, yes.
Or
And it could also be because when you have these capitals, right, and we don't find them there, the penalty is harder.
yeah, okay. So this is an appropriate liquidated distance, and substituting kata for CCA2B is actually
Not that.
is not that bad.
Okay, and a few more examples I just wanted to show, how sometimes the biologists are, maybe trying to make our life more difficult by using things like, the first time I saw OCT1, I thought they were writing a date. It turns out. And not only that, actually, there's also a number, and it kept on quite this thing again because this is a word, OCT is a word that they differ. The only thing they have in common is the annual number, so it does come fairly way down. Now, one thing we have to be cautious about is when we have acronyms in this field, there are multiple full forms of acronyms, right? Like AML could mean some kind of leukemia, it could be a name of a protein, it could be various things. So if it is not defined in that particular abstract, you have to be very cautious about it. So I just have one example we can't apply entirely. In those cases, we believe, hopefully, it will be the context will help us. So before I move on to the context, so let me say, these are the kinds of name internal clues we were looking at. And what we did, this line here is for the example-based method, and we applied it only when something was not tagged with F terms. So that's why it's so low down, the recall is only 120. But you combine these two, you can get recall in the 70s without sacrificing much on the precision. So there's a lot you get from name internal, but not everything. Particularly things like acronyms and so on, you're still going to have problems. So we started, well, it's typical then to, it's quite well known, you can use context to help this everywhere. It's sort of like thinking, take all names, think of it as one word, and then in our case, it has 23 possible senses, so you're doing sense disaggregation. And so the decision rule templates we use either a word or a bigram, 2 words next to each other. Then the other feature was direction, does it appear to the left or to the right? and the distance. And distance is always less than equal to some number. And some example rules that we learned were if you have proliferation of appearing to the left, then the name was a cell. It's a cell name. Or differentiation appears immediately to the right, as the last example here shows. Well, only cells get differentiated, so we figure that out here. Organisms, if you have infected with appearing to the left, it's like mostly a virus actually, or bacteria. And proteins, for example, if the word interacts appears immediately to the right, then it's a protein. And remember, in one of the earlier examples on co-reference, I said it interacts with something, so there we can actually figure out it's a protein.
So you're not doing any normalization of these strings, so interacts is different from interacting or can interact. Yeah. It's just on that string basis here. You were doing this interesting thing of reference merging, so if you have it followed by a context, you somehow feed that back to a context that you referenced. So, I'm sorry, you said it interacts with.
Yeah, there was an example early on where in a motivation slide I said, for co-reference, there was an example where the pronoun it was used, followed immediately by interacts. And I said, if I knew its type was protein, then I could figure out what the previous and for each expression was.
So my question is, do you do the merging before or after you do the classification?
No, this is only classification.
Okay, so they're staged in that way. I'm sorry, I thought you were saying something else which seemed interesting also, which is that suppose you have some phrase, some name, and you don't know whether it's a protein or something else. Then later you have an it which usually refers to it.
Yeah.
Based on hot heuristics or something. And that it is in the context of it interacts with. So if you did the merging first, then maybe you would get a context, an additional context which would give you a decisionless queue for the original word.
Right, but we are actually, a couple of things. One, we haven't built the co-reference module yet. And in fact, part of our motivation for doing classification was that it was going to have a co-reference.
Well, it'd be nice if they could interact with it back and forth with one.
Yeah. But we haven't really built the co-reference module. Okay, so a couple of other rules. When the word when appears to the left, 60% cell. Now, when the word by appears to the left, protein 43%, other organic compounds 15%, and so on. Now, are these good rules or bad rules? Actually, I contend that they are good rules. Because if you remember, the original distribution said proteins were 40%, proteins were only 15%, right? So it's giving a lot of information by having a different kind of distribution. So if I did something arithmetically, I can see that this is very informative. Likewise, what about by? Well, proteins is about the same, but other organic compounds in the training corpus distribution, that was like under 5%. Now it's boosted up to 15%. Actually, we're also intrigued to see why this was the case, why the distributions were like this. Well, when is often used in this literature to kind of like say the experimental setup. So it was like when the cell was extracted or it was cultured. So that's why you get cell a lot after when. And by, it's almost because it's like a passive construction, and yeah, things that do something are often proteins, chemical compounds and so on. And that intrigued us further to say, well, if they are like agents of certain actions, then maybe they might appear quite a lot as subjects in sentences. And just to test that out, what I did was look at like left context word being although, whereas, while, the, because these are like, you're going to have the subject immediately. And in fact, we confirmed it was almost identical distribution. Except the whereas context shows up here. Okay, so then coming back to this contextual rules, how do they work? Well, a few things we noticed. There are far more left context rules than there are right context rules. And I have no explanation for this. The left context rules are significantly more reliable than the right context rules. We also, actually we started off by taking the bag of words approach, which is, why look at just one contextual word? Why not just take a look at all of them? And it just didn't fare well here. And I think the reason is, because as I said, there are about 35, 40 names per short abstracts, right? And so, if you take longer context and a few others, you don't know whether that context is telling about this name or a name which is just 3 words away, right? So we observed something that Yurovsky observed in his word census derivation, that very close words are strong indicators and much more reliable. And that is what led him, I guess, to thinking about decision lists or the bag of word approach. And in fact, since context was where we began doing this work, that's why we started looking at decision lists too, once we saw this. And how do the contextual rules work? Well, I guess my main observation is you can get a lot of good contextual rules, but they don't apply often enough. We have like hundreds of rules. They all look good rules, but they appear applied, once in a while. So we'll recall when we have high precision, when we want high precision, it's like 5-10%. Right? And if we want to boost our recall to go up to 30 or 40, our precision drops down significantly.
And this is using only contextual words, not
This is
defined.
only using. Right? So we want to take all of these independent sources, put them all together. In fact, that's the exact matter. Right? So how do we put them together? Well, we thought, let's just do a weighted sum of all of these. And we looked at a number of cases where all of these sources were saying exactly the same thing, and how many times were they right? It was about a third of the time. The next is don't disagree and right means that there isn't a disagreement between these. Some just didn't say anything, right? And that's over half. And there are more examples where they differ, but when I take the sum, I get the right answer. Example encoded by C false. Our example-based method said it was a protein, but there's the strong contextual rule that says encoded by appearing to the left means we're talking about a gene, right? Because only genes encode things, not proteins. And that goes around, and when you take the sum, we get the right answer. Now in this case, this is an F term mutants, but mutants can be different kinds of mutants. And so if you just go with mutants and take the highest one, that was, I guess, I don't even know what I guess, I'm not sure. However, again, the context said, since it's infected with, this is an organism, and it turns out, in fact, this is a virus, and it's a mutant. Now, here's an example where the context got it wrong. It's secreted, usually what follows is a cell name, but the suffix here, TAC, told us it's a protein, and so it had a high likelihood for protein, so when you take the sum, you got the right answer. So how do these answers look like? Well, without setting any limits to these, our precision was about 83, recall 82, and F measured 83, and we varied it quite a lot, and you notice the F measure hardly changes. This is like threshold of everything was at least 0.9. Right? And all the way to no limit of 0.2. And Of course, when you go higher, you get slightly higher precision, and the recall drops a bit. But, so that's how that looks like. So what are the other factors that I think are helpful? Well, coordination, my intuition is if you say something A, B, C, they're all likely to be the same type, A, B, and C. Right? Well, that happens actually about 76% of the time. Why does it sometimes differ? Because at a higher level of abstraction, this might work, like a protein and a protein complex. Right? Now these are different types, but at a higher level of abstraction, you can think of it as a single type, and that's that's the kind you find there. Or in this case, where you have a lipid with some proteins, well, that's functionally they're all doing the same thing, stimulating something else. Right? But if I just go with these types, it works about 76% of the time, and sometimes that might help. Right? Now, as I said, acronyms, detecting the full form is very helpful. Because if you do that, the full form, you can get the type of information from that often. And what we found was we built an acronym full form detector, just almost like spending a weekend on it. Nothing fancy. We believe a simple method will suffice for our purposes. Why? Because even if we don't get the left extent correctly, right? So you might have something that says mitogen activated protein kinase parenthesis NADK, right? Now I know that at least the right limit is kinase, right boundary is kinase, and that's what really helps us. Even if I didn't get the left boundary right, it doesn't really hurt us that much. Of positives, in this domain, I don't know whether I want to generalize beyond this domain. There's a lot of positives, and the positive phrase often gives you the type information, such as MEK1 or tyrosine kinase. So I can, get the type of this, noting a positive, it's in a positive, I can assign it to MEK1. So, what are we doing now? Well, there are certain kinds of things I really like to automatically extract, but I'm not sure how to do that. For example, headwords like clone, mutant, family, they get distributed over different types, right? But it's almost like if I say cell, clone, if I knew the thing to the left was a cell, then the whole thing is a cell. Right? And all of those are exactly the same type. Right? They don't give you the type, whatever is to their left gives you the type. In some sense, I don't know what to think of this is the modifier of the thing to the left. So it's sort of like clone off something. And how to automatically detect this kind of thing, you don't know.
It's the reverse of specifier. Specifiers a particular kind.
Right, exactly. And the specifier is the next one. It's actually, we found, I showed you an example where protein followed by an Arabic number means it's protein. And actually, it's far more general. You can take any family, X family, put in this number after that, you get a particular instance of that family. I don't know how to learn that. Automatic, some kind of automatic method. Looking at the context, I'm kind of like disappointed at the recall for how much we were able to do. And I began to wonder, is this really because of the data, the kinds of abstract, is it because of that, or did we not have sufficiently large annotated corpus? So one thing that we plan on doing is take our really high precision rules, sort of think of them as the seeds, like in Yurovsky's bootstrapping method, take those as seeds to classify more names, and then iterate this process. Go ahead. Other questions I would like to work on are, we just did a simple, on our own, decided how to weigh these different things. really like to learn how to set weights to the different things. One thing actually began to wonder whether we should have even considered a decision list at this point, because you notice the way we set up our templates, whether it's for context or even name internal things, what you do have is, two rules where one condition could just say if the last word is protein, and another one could say protein followed by a specifier, which is a number. Right? In these cases, when you have such, where one rule's condition subsumes another one, what should we use? I mean, addition is the standard method is to use, whichever gives you more information, but that doesn't I'm not sure that's the right way to do it, because I think you have to take the more specific information, right? And the more specific it is, the better, except of course you're going to have, more specific means you might have less examples and less training data. So if you're willing to, if you do some kind of smoothing for that, should we use that? Sort of like saying, okay, maybe I shouldn't even use these as decision lists, I should use them as decision trees. After all, decision trees, we're going to go all the way down to the loop. Okay, so a positive is, an adjustment for frequency. Our rating right now doesn't take into account the frequency, and obviously that's something we have to do, so we haven't done that yet. Let me kind of quickly end up with, say, two things that we probably would like to do as post-processing. And that is, when a name appears, sometimes it often appears several times. And supposing, and it often does occur, that we do give different types for the different occurrences. And I thought of, again, taking inspiration from Jurowski, say, we had 1 cent per document constraint. Are we going to have the same name with different types? Probably not. So why not try one type per document? We're going to try that out. That's something I'd like to try out. And I don't know how exactly to apply coordination. As I said, it often helps you. And should we do it only, like, for example, when I have A, B, and C, and a new, say, from the context, the type for C, and I didn't know, have a good guess for B, should I do it only then? Or should I do it right from the beginning? And so those are the kinds of questions we're still investigating. In some sense, just to emphasize, this work is more, as I see it, is investigation of the features that kind of help rather than the actual method in which we put these together. And a couple of things I would like to note is this domain suffixes are really helpful. F terms, I like to think of them as saying it's a word but also positional, right? Unlike, say, Mike Collins' work on named entity classification, where all he used was words that were contained in the names. And I think here it helps to know the position. And then when we want to do some kind of similarity-based method, we need to normalize and include some kind of right bias. That's one of our conclusions. As I said, a lot already about contextual, that names appear close to each other, so only really local name words are helpful. For more data help, and so on. Acronyms, we believe, are very crucial to detect acronym full forms. And positives are occurring quite often enough to be helpful to do that. Okay? Thank you very much.
How did you discover the features like the site, SYT, etc.? Is this done by hand, or did you use visual information or something to decide what features are indicative of the type?
Well, initially we were just looking at, say, for example, site. How often we look at the distribution.
It's done by inspection, is that right?
No.
I mean,
No,
well,
we
how
don't.
was the word site, or not word, how was the string site selected?
Oh, because we consider all possible strings, right? Suffixes.
Starting with all three-letter suffixes.
We start with 3 letters. We try to decide, look at the distribution. We decide whether we should go one more to the left to see if there's gain in information.
That's what I said, so you use some mutual information function.
Right, yeah.
Yeah, okay. And as far as, well, there are other things, right? Like something is on the left, and there's a list of things that you're interested in. So all this was discovered with the help of some kind of mutual information function.
Right.
Yeah.
So with respect to the use of suffixes and the use of both word internal and external features with bootstrapping, and with respect to some of this, but not all of the smoothing questions that you raised, you might take a look at Sylvia Kucharzan's thesis, which she defended earlier this fall, and at his other work with as a student of his. Since I think you guys have been thinking along very similar lines of these. There's also work which is related to the bootstrapping stuff that's been done by Alan Ryloff, for example, and Roman Yednover. So I can give you these references, but I can mention that for some other people who might not know that stuff. Also, I was interested in your use of conjunctions as a clue. I think actually I've seen some technique like that before for some other problem, but I can't remember where. It might have been Sharon Caraballo's work. But there's a nice way, I think, maybe to integrate this into decision lists, where you were asking, well, you know, which thing do you disambiguate first, and when do you apply this rule? And if you put all of your decisions together into the same list, then it may be that one of the features for deciding B is that you know C and C is nearby, or conjoined or in the same document or whatever, and that may override other contexts that show up for C. What you end up with is something that looks like a transformation-based learner, where you have a bunch of different cues, and at some point, the fact that you've resolved this one can continue to resolve that one. Might be one way.
Right. One thing I forgot to mention when looking at the context rules, the results that I had shown that pretty low recall. We tried, we thought, well, left context and the right context are completely independent of each other, right? So we can keep them as 2 separate lists. Now the question is, how do we put them together? We thought of 2 simple, obvious ways. One is, if I had one single list for context, then I would go with the one which is the highest, right? So we tried that. Or we could try something which said, if one of them said it and the other didn't, take that. If both of them did it, kind of like take their average. And in fact, I think that up to, let me, if I can quickly get to that. Oh, sorry, I went way back. Right? Those 2 lines are almost on top of each other. It doesn't seem to make a difference at all. However, I'm trying to do the same thing with context, and there is a difference. It's better to keep them separate and take some, rather than say go with whichever. So I'm not sure I want to put them all into one single issue.
Another reason to keep them separate is then you can do code training, which is what somebody did.
Yeah.
In some instances.
I'm wondering whether the reason that the left context is more useful is it's just a standard for subcategorization, and there's stronger constraints. So I was just wondering, if you look at the kinds of rules that you have, do you get lots of verbs, as you said, which are saying what their object has to be?
That is intriguing. I haven't even looked at what the context rules, which ones in right or not good or the left as good.
And I'm just wondering if you said you were sort of disappointed with the recall, and I was just wondering if you had whether you think that you would do better with a contextual kinds of rules if you had a better parse, if you had a parse at all. I mean, well, sometimes it might not make that much of a difference, but,
Yeah, well, actually, although we have never done parse here, I mean, one of the main reasons we did this, got into this, was for information extraction. Although we don't build a parse, we do some simple NP chunking and word group chunking and so on. And maybe if I use that information, since we do that anyway, and it's not very costly to do that. Maybe it might.
right, even doing part of speech tag and identifying verbs.
And just following up on Bob, but it seems like if you might observe that there are common patterns of relationships between like proteins and cells, so that even if you don't recognize the verb that's necessarily connecting the two things, just because the cell is in subject position and the protein is in object position, then you can, and something's in object position. You can assume, so it's like instead of that.
Oh, you're saying like, even without looking at the verb, look at subject object.
Yeah.
Right. So, I mean, these are just more of your conjunction features.
Right, more elaborate.
These sorts of things, depending how deep you get into the syntactic analysis, the sort of stuff that's been looked at in information extraction, that they can't land, if I remember correctly. Patrick Mantel, I think, had one example of that sort of thing, where you're going with that are variations of the paraphrase problem, right? You're looking for a variance, regular patterns of variance. To the extent that you're going to parse this stuff.
Right. Well, actually, we would take off the shelf, NP chunkers, or for part of the stagger, we could rule stagger. Now we use the FFT stuff. And we use that for our base NP chunking too. So we overlap to confidence, I mean. But they work fairly well, actually.
That leaves the question, for a domain like this, you're telling us that like part of speech tagging pretty much pours without much effort. But what would a good parser do on this? What would Charniak's or Collins's parser do to catch things like MK1?
Well, things like Collins, which are quite lexicalized, I guess most of these verbs one. You probably have to train into these domains.
Well, but maybe you can use endings to determine that something's a verb.
I'm sorry?
Then maybe you can use either endings or classification of the common words or something like that to be able to change your biases.
Right, but the kind of sub-categorization information that is used, it's not that, unless
Verbs ending in HEE in the biological domain are probably transitive.
actually,
Maybe
I would say most of them are probably transitive.
most verbs in general.
Or maybe I should say interaction kinds of verbs, because they're talking about interaction.
If my intuition is backwards, rather than thinking that parsing will help you do the categorization, I feel that perhaps you can chunk the NPs far more reliably and categorize them and use that to parse. We can abstract out the lexical item.
Well,
and replace it.
that is exactly what happens for us in our next level, right? Knowing the type and knowing is our pattern. What we do is do simple like base NP chunking, verb group chunking, and so on. And then we might have patterns, but those patterns look for not just a noun phrase, but a noun phrase of this class. And it's almost like building a semantic grammar